Recording, and I'll turn the time. Good morning, everybody. Welcome to the Utah Centers of Excellence in Hematology virtual seminar series. I'm going to turn the time over to Anna Bowden, who is actually hosting our speaker for today. And thanks, Anna, for taking the time to do this. And thank you, Kyle, for being willing to speak to us today.
Okay, awesome. Thanks, Diane. It is my pleasure to introduce my friend and colleague, Kyle Hewitt, who is an assistant professor in genetic cell biology and anatomy in DGCA at UNMC. Kyle has a long career in studying stem cell regulation and differentiation. It started where he studied fibroblast differentiation from iPSCs at Tufts as a PhD student. He then joined Emory Bresnik's lab, where he studied GATA2 enhancers as regulators of stem and progenitor behavior in hematopoiesis. And this discovery led him to discover a novel gene, or gene family, I guess, that he's going to talk about today. This is the clue that he's going to just talk about that regulates both erythropoiesis and hematopoiesis. I guess today you're going to talk about regenerative erythropoiesis. Is that correct, Kyle? Okay, and yes. I love this work, so I'm very excited to hear what your next steps, or I get to hear Kyle's work a lot, so I'm very privileged, but I'm excited to hear what you have to say today. Please, let's hear what you got, Kyle.
Well, thank you, Anna, for that nice introduction. And thank you for the invitation. Excited to speak about our ongoing work today. Some of it is very recently published, and some of it I don't think Anna has even seen yet. So hopefully this will be interesting for this group. So is that advancing? Hold on. There. Okay. So really, two main parts to what I'd like to talk to you about today. In the first part, we're using some single-cell transcriptomics data to try to understand new mechanisms of stress-initiated transcription in erythropoiesis using an acute anemia model, and trying to identify some of the immediate transcriptional changes that are required for the acceleration of erythropoiesis and anemia. And then in the second part of the talk, really getting into some more mechanistic work on this protein that we've been studying since I was a postdoc on SAMD14 and how it's controlling mechanisms of erythroid signaling in stress contexts. So it's not lost on this group that there's a progression during erythropoiesis through a series of progenitor cells that produces the mature red blood cell. In the context of regeneration, for example, if there's an acute loss of red blood cells due to bleeding or cell lysis, it requires activation of progenitors to meet that demand, to actually exceed physiological erythropoiesis and restore homeostasis of the system. And as it turns out, this occurs through a unique population termed erythroid stress progenitors that are separate from the normal megakaryocyte erythroid progenitors that's actually contributing to the production of red blood cells in the context of acute anemia. What's poorly understood is what are the mechanisms that are driving the progenitor function in the context of regeneration. And it's sort of our lab philosophy that when we annotate and understand these mechanisms that are augmenting erythropoiesis and anemia, we can discover new mechanisms and potentially new diagnostic or therapeutic markers in hematologic disease. So this project kind of began from our understanding of cis-regulatory mechanisms that are regulating hematopoietic and erythropoietic development. Some of the sort of famous genes that are regulating hematopoiesis and erythropoiesis are controlled by a cis element termed a composite EBOX GATA element. These elements are occupied by TAL1 and GATA, either GATA1 or GATA2, in addition to some additional components of a multi-factor complex that's controlling enhancer activity at these key hematopoietic genes. So what we've conducted is a kind of multifactorial prioritization of dozens of these EBOX GATA elements throughout the genome to try to identify new cis-regulatory mechanisms that are regulating erythropoiesis and try to annotate them into what they might be doing to promote either normal embryonic development, regeneration of tissues and cells, like we see with some of the elements that we've been studying, or potentially in carcinogenesis as well. And so the focus of the talk today will be on sort of cell and tissue regeneration mechanisms that are controlled by these EBOX GATA elements, and what we've discovered with this prioritization approach. And again, our hope is that by prioritizing these elements, we can identify new mechanisms regulating the regeneration responses in acute and chronic injury. So one of the elements that started this whole project off was the SAMD14 enhancer element. This is an EBOX GATA element very similar to the GATA2 plus 9.5 element that's required for hematopoiesis. It's located in intron 1. It's occupied by, let me get a pointer going here. It's occupied by GATA2 and TAL1 in intron 1 of the SAMD14 gene. We knocked it out of the mouse genome in order to identify the role for this intronic element in controlling either hematopoietic development, homeostasis, or regenerative mechanisms. In the steady state, this cis element is not required for hematopoiesis or erythropoiesis. The mice are perfectly normal. However, it is strongly transcriptionally active in CMPs and MEPs in particular. This cis element strongly controls SAMD14 expression. And this is just showing sort of our wild type versus our enhancer knockout bone marrow sorting for his CMPs, GMPs, and MEPs. You can see that this enhancer knockout reduces SAMD14 expression. In the context of acute stress, if we treat mice with the red blood cell lysing chemical phenylhydrazine, almost all of our enhancer knockout mice die. And this is a dose it's a pretty severe erythropoietic stress, but all of our wild type mice, while they get very anemic, they are able to recover over this 7-day time course, whereas our enhancer knockout mice are not.
Sorry,
indicating.
Kyle,
Yes.
sorry. You have a question from Vanessa. Vanessa, do you want to ask that, or I can either way?
Yeah, sure.
Go
Thank
ahead.
you. Thank you so much. It's a great talk. Thank you so much. I'm super excited to hear the rest of it. I just had a quick question about the SMAD14 expression in the different populations of progenitors. And if you also looked at an erythroid committed progenitor a little bit downstream of the MEP.
Yeah, so yeah, I'm kind of skimming a lot of data here, but yes, it's definitely regulating erythroid precursor SAMD14 expression as well.
And just to clarify, this murine MEP, what was the immunophenotype of it?
It's that
Is it from the Pronk scheme?
Is it
the pre-90?
Yeah, FC receptor low, CD34 low.
Okay.
Right?
Thanks.
Yeah. Yes, so thank you for that question. Yes, so it is beyond just the MEP. It is also downregulated in the erythroid committed precursor cells. And then the other thing that we sort of linked it to, and I guess this is kind of obvious at this, from what we know now, but we really didn't have a good sense for this, but SAMD14 is elevated in the context of acute anemia in wild type mice. Its expression is going up in CD71 positive, TER negative, KIT positive cells. It's that transcriptional upregulation that's prevented in our enhancer knockout. And that inability to upregulate SAMD14 in acute anemia is resulting in their failure to recover. And this upregulation is also happening for the transcription factors that are occupied at that cis element. So GATA2 and tau1 are also increasing in expression in acute anemia. So we wanted to know what else was changing at the transcriptional level in these acute anemia models. And we decided to do this in single cell sequencing samples. So, and control versus anemia mice treated with phenylhydrazine for 3 days. We took out the KIT positive cells from the spleen, where the stress erythroid progenitors are being activated in acute anemia. And we conducted single cell sequencing to allow us to look at not only transcriptional changes in the entire population of KIT positive cells, but the heterogeneity of that response within different subsets of cells. Some of the questions we're asking with this data set is really how prevalent is a mechanism like this SAMD14 enhancer mediated transcriptional upregulation in regeneration? Can we use known omics data to predict the control of anemia activated genes by enhancers that have similar characteristics to the SAMD14 enhancer? And then what are the kinetics of transcriptional activation over a time course of anemia recovery? So not surprisingly, we see the erythroid subsets of cells within our single cell transcriptomics data are expanding in the context of stress erythropoiesis. So this was kind of expected. What the single cell data allows us to do now is to really take that variable out of the equation and kind of ask, well, within that subset of cells, what are the transcriptional changes that are occurring irrespective of changes in cellularity? And so we really wanted to assess this data in 2 different ways. One was whether the transcriptional changes were happening within the subsets of sorted cells identified by their transcriptional profile, and whether the transcriptional changes were also happening as a result of changes in cellularity. So if we look within the CD71 positive cells, CD71 positive KIT positive cells, at the SAMD14 expression in each cell, we see statistical increases in really the number of transcripts per cell. What we also see is that within 5 distinct erythroid clusters of cells, we see increases in the percentage of cells within those clusters that are expressing SAMD14 transcript, more than 1 transcript per cell. So this data really suggests that it's both. We're seeing increases in the cells expressing these stress activated transcripts, and we're seeing in a per cell basis that the transcripts per cell is increasing. So we also found that within these 5 distinct erythroid clusters of cells, we see that anemia is inducing transcription within specific clusters of cells. So you can see here, I'm showing you some examples of some erythroid genes that are going up specifically in this E3 cluster. Again, that's both at the number of cells expressing it as well as the expression level per cell. So in order to identify the changes in transcription factor activities that were happening within these clusters of cells, we turned to this bioinformatics tool called CIHD. CIHD takes the transcriptional information of the transcription factor on a per cell basis and links it to its target genes within that same cell. And it does this really based on two fairly simple parameters, just co-expression and the presence of the cis element that it would be expected for the transcription factor to bind at that target gene. And so it's a fairly simplistic analysis on sort of an individual gene basis, but multiplied over all the cells and all the transcription factors and all the target genes, it provides a very powerful tool for us to begin to predict what these where these transcription factors might be active within these subclusters of cells. And the pattern that we see emerging from this analysis is that this erythroid cluster 3 is very active within the GATA1 and Tau1, as well as some of the other transcription factors that are known to co-occupy chromatin with GATA1 and Tau1, including LMO2. And so we see this co-occupant, these transcription factor activities are high within this subset of erythroid cells, and sort of leading us to believe that maybe this is a particularly active population of cells for these transcription factors that are regulating SAMD14 expression.
Sorry, Kyle, will you go back one slide? What's the 1, 2, 3, 4, 5? I missed it. Is that?
Yeah, so they're distinct clusters of erythroid committed cells identified with our single cell sequencing data.
Just because that 3 is so strong, I just was trying to figure out relative to 3 what the others were. And it looks like Vanessa also has a question.
Similar question. I'm wondering, like, if those 5 clusters are just different stages along the differentiation process, and if like maybe you did like some pseudotime analysis of the single cell RNA-seq data or something like that to kind of get a clue for like the relationship between these 5 erythroid clusters.
Yeah, so we've done that. The pseudotime analysis tells us that this is kind of that this erythroid 3 is kind of middle to late erythroid. It's less than perfect. It's not been a very satisfactory analysis because it doesn't give us, like, you see the kind of simulated data where it shows a really nice trajectory, and we don't really see that in our data. But yes, it does suggest that kind of the erythroid 1 cluster is probably less differentiated. That one's expressing sort of metalloproteins. Erythroid cluster 4 seems to have higher expression of the hemoglobin genes, suggesting that that's later than the E3 cluster. So this is kind of in the middle of these 5 clusters, but it doesn't, I don't know, it hasn't been a very satisfactory, at least the way we're doing it. I don't know if you've done those analyses before, but it really depends on where you set your baseline, right? So you have to pick a cell that's like your HSPC, and we can define those, and we see those pretty well. They express very high levels of GATA2. But within that cluster, you move it around, you get a completely different trajectories. So I don't know, it's frustrating.
So then I have another idea, but maybe it's like kind of one of those, like, trying to like reach all the way around your head to touch your nose things. But if maybe taking your single cell RNA-seq data might help you identify some unique cell surface markers or something like that, where you could then like pull out these cells from each of these clusters and at least just do like a cytospin or something to look at their morphology to sort of see if you could try to stage them that way.
Yeah.
Seems complicated though.
No, and that's a great idea. We put that into grant applications to do that experiment. And there are definitely cell surface markers expressed specifically in one cluster or another. They tend to be, at least the markers that are specific are ones where either there's no antibody or or a poorly characterized antibody, it's probably a whole thesis project that I need to talk a student into doing to try to identify. I mean, it's a dream of mine to be able to identify functional distinctions between some of these different clusters of cells so we can link to transcriptional activities.
Cool. Thank you.
So the next step here was we had to deconvolute the data, but we wanted to, because the prior analysis was just all of our data, control and anemia conditions. But if we look at the differences in these, they're called regulons, but it's really an activity score for each transcription factor. We see within that same erythroid cluster 3, we see a very strong increase in the predicted activity score of GATA1 and Tau1 in response to anemia, suggesting that anemia is inducing increased activity of GATA1 and Tau1 in this specific erythroid cluster 3. So at this point, we wanted to develop kind of parallel strategies to predict how prevalent the SAMD14 enhancer mechanism is during erythroid regeneration. And so we use sequence determinants. So we wanted to look for sequences that had that E-box with a spacer in between and then a GATA motif as kind of a substrate for binding these transcription factors. We used GATA and Tau1 occupancy using available ENCODE data sets, chromatin accessibility by ATAC-seq, and then, of course, whether they're transcriptionally activated in anemia. So we took all E-box GATA sequences. There's 80,000 of them in the mouse genome and about 100,000 in the human. And we asked what the ratio is of these E-box GATA elements annotated to nearest gene. What is the prevalence of anemia regulation at loci that contain E-box GATA enhancers? And if we compare all detected transcripts in our data set to just the anemia upregulated transcripts, we see that E-box GATA sequences are enriched at anemia upregulated gene loci compared to all of our detected transcripts. If we then filter that data based on the occupancy of GATA or Tau1 at these loci, we see further enrichment of the ratio of anemia upregulated transcripts that can be found nearby these E-box GATA elements. And so this gives us some confidence that this may be a fairly prevalent mechanism for regulating transcription in the context of anemia. We see within this erythroid cluster 3, we see about 65 transcripts being upregulated that are nearby these E-box GATA elements that are occupied by GATA and Tau1. And so we selected a few of these for further analysis. So the SSX2IP was one of the highest upregulated transcripts that we found in our data that had an E-box GATA, a nearby E-box GATA enhancer. And then this other one, TTLL12 as well, that we selected for further analysis. And we were able to confirm by sorting and just conducting qPCR that these two genes are strongly upregulated in the context of anemia. So here we're sorting CD71 positive, TER119 negative cells from the spleen. And then if we look at the nearby E-box GATA element that has open chromatin accessibility and GATA occupancy, we conducted an analysis called FAIR, which is slightly a variation of a chromatin accessibility assay. I think a lot of people use ATAC-seq just regularly now for everything, but this is an assay that I've used for years to look at changes in chromatin accessibility using formaldehyde-based enrichment of open chromatin regions. And we can see in anemia, we see an increased accessibility at the SSX2IP and TTLL12 ebox-GATA sequences in response to anemia. So suggesting the genes are going up, but also the chromatin at the nearby candidate enhancers are also increasing in accessibility. So what is SSX2IP? This is a pretty poorly studied gene. Its expression seems to be highly specific in the hematopoietic system, seems to be highly specific to the MEP and downstream erythroid cells. If we look at kind of published RNA-seq data, the known mechanistic information links SSX2IP function to the structure of centrosomes and the stability of chromosome segregation in S phase. And there's a few papers that have shown that high expression of SSX2IP is linked to leukemia cell proliferation. The candidate enhancer that we found was in intron 1 of the SSX2IP gene. Here I'm showing you some ChIP-seq data, just showing that H3K27 acetylation, GATA2 and Tau1 are enriched there, as well as it's in an open chromatin accessible region. So what my graduate student Yichao did is he designed a couple of CRISPR-I constructs. So these have the DCAS9 CRAB domain that can be targeted to chromatin using guide RNAs. He also he targeted it specifically to the enhancer region. He also generated a control that targets the same intron, but just at a distinct site. And he asked whether he could disrupt the activity of the enhancer. And so here's that data shown in K562 cells. So here's a scrambled control expression of SSX2IP. If he expresses that intron 1, he calls it the non-targeted control, but he sees a slight reduction of SSX2IP transcription, but that's greatly reduced when he expresses the guide RNAs that are specifically targeting the enhancer of SSX2IP. And the same result was seen targeting the TTLL12 enhancer as well. So this validates our prioritization approach. It tells us that we can sort of use these criteria to identify functional enhancers that are regulating genes that are upregulated in the context of anemia. He also used that same chromatin accessibility assay to ask whether the DCAS9 CRAB targeted guide RNAs could reduce chromatin accessibility, and indeed they do. If we compare the non-targeted to the enhancer targeted, we see a reduction in chromatin accessibility at SSX2IP and TTLL12 enhancers. So what does SSX2IP do? So he conducted some knockdown experiments where he lowered SSX2IP expression with an shRNA. And this was in mouse primary spleen cells that had been treated with phenylhydrazine for 3 days in vivo. So these had been stress activated. These are stress activated progenitors, then taken out of the mouse and grown ex vivo to look at erythroid differentiation. And so he can efficiently knock it down with shRNAs in erythroid committed cells. And as you can see from these, from the CD71 TER119 flow cytometry analysis, we see fewer of the undifferentiated erythroid cells and more of the differentiated cells. And so the SSX2IP knockdown is increasing the percentage of more mature erythroid cells in these cultures.
Kyle, just for a question on that knockdown, is the knockdown done in vivo and then you harvest the cells, or are you doing the knockdown in vitro?
No, we're doing that in vitro. So we take the cells out, we lineage deplete, and then we we infect them with the shRNA containing virus.
Thank you.
Yeah, so then here's just the quantitation of that data, just showing, again, so I guess it's a 3-day culture, right? So we take the cells out, we infect them, we culture for 3 days in just SCF containing, EPO containing media. And again, the cells are more differentiated in the absence of SSX2IP. We also we spun the cells, the GFP positive cells, down onto slides, and as is fairly apparent from just the morphological analysis, these cells are smaller. We can quantitate that just on the forward scatter channel on the flow cytometer to see that the knockdown is reducing the size of these erythroid precursor cells. We also looked at colony forming potential and showed that after knockdown, we see fewer percentage of KIT positive cells and fewer BFUE and CFUE colonies after knockdown. And then finally, we're just looking at what's known about SSX2IP function. And it had been linked to kind of the centrosome stability and cell cycle progression. And so we looked at that by incubating cells with BRDU and using an anti-BRDU antibody with DAPI to look at cell cycle. And what we found was a shift in the percentage of cells that were in the G1 versus S phase of the cell cycle. And there were actually an increased percentage of cells in G1 and fewer cells in S phase, indicating that knockdown was altering the rate of progression through various stages of the cell cycle.
Kyle,
Yes.
can I ask you a question to clarify, because I'm getting a little confused?
Sure. Yeah, I'm rushing through it a little bit.
No, you're not rushing through it. I get easily confused. So because this gene, I mean, you're pulling this out in the context of like regulators of stress erythropoiesis, right? But you're getting more mature cell differentiation, right? I guess one of my questions would be like, so but you're showing this as a frequency of cells. So I guess if you would do like total quantification, because this is ex vivo, not in vivo. If you would do like total quantification overall, you would get far less because you're getting far fewer progenitors. Is that the idea?
Yeah, I mean, we don't have, as kind of Diane was indicating, we don't have the in vivo function of SSX2IP. This is an ex vivo assay where we've sort of normalized the percentage of Lin negative cells and knocked down SSX2IP.
Right.
And so there's several possibilities here. I think one is that it may have distinct roles in the progenitors versus the downstream cells.
Right.
And this alteration of kind of the rate of cell cycle progression may have distinct effects on different populations of cells, as Merav Sokolovsky has so beautifully shown with her work. But we haven't gotten that detailed yet with the types of analyses that we've done with this particular gene. This is really kind of I think, setting the stage for a larger project.
Okay, cool. Just wanted to make sure I wasn't missing anything.
No, you're right. It was very kind of I guess annoying at first seeing that data and being like, wait a minute, why are we getting more cells?
Right.
More of the cells that we you would think that the system should want to produce more erythroid cells. It could be a stem cell exhaustion thing or something related to kind of the role of SSX2IP in either the initiation event of stress erythropoiesis, the expansion events, or the kind of really the later differentiation events.
Okay. All right. Great. Thanks, Kyle. Appreciate it.
Sure. Yeah, and that really leads me very nicely into our current model. So in the physiological context, SSX2IP is expressed in erythroid cells, and in other contexts is known to promote centrosome stability. In the context of anemia, this EBOX-GATA element is activated to upregulate SSX2IP expression. And we've shown using shRNA knockdowns that in the context of acute stress, the SSX2IP gene is promoting progenitor activity and altering the ratio of cells in the culture that are at different stages of the cell cycle. I will stop for questions here. I'm seeing something in the chat.
I think you might have answered that question.
Yep, I think we got them answered.
Okay,
Yeah.
good.
I think immediately you put those data up. Yes.
Okay. All right. So in the last 10 minutes or so, let's see how quickly I can go through the second part. All this work really led us to kind of this interesting question about kinetics. And so we've done some time course analysis now of the different stages of anemia. So we pulled out single cell data from day 1, day 3, and day 7, and looked at the changes in cellularity and the changes in transcription that's occurring within the same clusters of cells and in distinct clusters of cells at these different time points. What this data always tends to show with these big data is that we probably need more time points now because this data is extremely interesting. We're really excited. It spawned many new research directions. You can see that some of these clusters of erythroid cells are really not activated at these early time points. Day one mice are anemic, but you can see that they haven't quite let that anemic time point has not quite led to the expansion of the erythroid cells. So this is still kind of the activation stage of erythropoiesis at day 1, whereas at day 3 and day 7, you really get a lot more of the expansion of those erythroid cell populations, and leading to the ultimate resolution of anemia. So just to give you a sense for what we're seeing in this data, we see a lot of dynamic gene regulatory patterns that are happening, and perhaps we could predict this, one of the reasons why we did this experiment, but it's really cool because it allows us to separate out the transcripts that are associated with distinct transcriptional states, right? So we see, for example, we see these 3 different states, state 2, 5, and 8, that are activated very early. So these genes come on at day 1. Sometimes they stay elevated for a period of time, but oftentimes they're also rapidly deregulated. So between day 1 and day 3, we see a rapid downregulation. And again, this is within, if you look at this state 8, for example, these at day 1, these cells have not expanded yet. So these are definitely genes that are priming the erythroid response. We see other states, and this is actually the state, all the data that I just showed you, this sort of middle stage activation state. So these genes are on at day 3, but for the most part, around 85% of them are not on at day 1. So these are kind of the middle stage activation. So those EBOX-GATA genes are generally associated with this kind of middle activation state. We also see a late activation state. I have no idea what these genes are doing, but at day 7, these genes are turning on. At day 7 is the time when we see the resolution of the hematocrit and hemoglobin and red blood cell count levels back to normal. So these mice, in terms of their red blood cell physiology, they're going back to normal, but these genes are, there's a subset of genes, thousands of genes that are turning on at these late stages. And then we see a repression state as well. So again, we're really interested in kind of digging into this data and trying to identify what the transcriptional mechanisms might be and how those transcriptional mechanisms might relate to distinct stages of the regeneration response in stress erythropoiesis. And sort of to put that into a model, here are kind of the blue line is showing the expression patterns of our S14E-like, our SAMD14-like enhancers. So these are turning on at 3 days post phenylhydrazine. This is a stage, if you're looking at the hematocrit here in the black line, this is when the mice are generally most anemic. And then by day 7, they're recovering. And so we're really interested in kind of moving forward in identifying what these different waves of transcriptional activity are doing after stress regeneration, during the stress responses, and we think that they're linked to mechanisms that are required for the regeneration of the erythroid system. And I think I've mentioned all of these points already, but we have kind of a cohort of these EBOX-GATA associated genes upregulated during recovery. The SSX2IP gene is a candidate for regulating the cell cycle during the erythroid stress responses. Some of these genes are known to be misregulated or mutated in hematologic disease, but we know very little about the mechanistic relationships between the transcriptional activation and what they might be doing to promote erythropoiesis. Yeah, I don't know that I have time for this, but we have a whole other project ongoing in the lab looking at mechanisms of SAMD14. I think this might only be 5 slides. So if you want to give me 5 minutes, I can whip through it because I think some people in the audience might be interested in this part. But we've previously identified a multi-component mechanism that's regulating SAMD14 activation in stress. And in the absence of this SAMD14 enhancer, as I described earlier, anemia response cannot activate this EBOX-GATA element. SAMD14 expression remains low, and that results in lower output of KIT signaling. So if we look at phosphorylation of AKT as a downstream indicator of KIT signaling in response to SCF, SAMD14 is driving higher phospho-AKT and higher phospho-ERK activation in response to SCF, right? And so we developed this rescue model to a genetic complementation model to add back SAMD14 or mutants of SAMD14 that might be regulating SAMD14's activity. So again, this is similar to the assay that I showed you. We treat mice with phenylhydrazine. 3 days later, we isolate spleen, lineage deplete, and then we can infect them with a SAMD14 containing retrovirus and do flow and colony assays. And so we use this to show that we can restore signaling through the KIT receptor by adding back full-length SAMD14, right? So then we're able to look at various domains of SAMD14 to see if they're required for SAMD14's activity. The first candidate was the sterile alpha motif domain. This particular domain is well established as a protein-protein interaction domain. So proteins with SAMs often homodimerize or oligomerize and form these long polymers sometimes in cells, although sometimes they're just forming dimers and sometimes they don't interact with other proteins at all. So we wanted to test whether the SAM domain of SAMD14 was promoting erythroid activities. And we showed here on the left that the SAM domain of SAMD14 was promoting colony BFUE activity. So if we express the full-length SAMD14, we can increase BFUE activity. But in the absence of the SAM domain, we get fewer BFUE cells compared to full-length. Also looking at cell survival using an Exin-5 and DRAC7 DNA dye, we can see a strong pro-survival effect by expressing SAMD14. right? So this here's our empty vector late apoptotic cells. So these cells would be an Xn5 positive and DRAC7 positive. We see a strong pro-survival effect when we express SAMD14, and at least partially, this effect is conferred by the SAM domain. Although this is very clearly just a partial rescue of SAMD14's the delta SAM version of SAMD14 is only conferring part of that pro-survival effect that we see by expressing full-length SAMD14. Next, we wanted to know what SAMD14 interacts with. We did an HA pull down of the full-length SAMD14, and the top candidates that showed up on this interactome analysis was this family of actin capping proteins. So the beta and alpha subunit, CAPZB, and alpha-1 and alpha-2 were by far the top 3 candidates for SAMD14 interacting proteins. We were able to validate that interaction by Western blotting, showing here in this middle lane that when we immunoprecipitate SAMD14, we also pull down alpha and beta subunits of the capping protein complex. However, what was surprising here is that we were also pulling this capping protein complex down in the absence of the SAM domain. And again, the SAM is protein-protein interaction motif. We just assumed that this was going to be an important domain for conferring this interaction, but it's completely dispensable. And so this capping interaction was happening independent of the SAM domain. So what does this capping protein complex do? It had not been studied at all in the context of hematopoiesis, but actin capping is extremely important for overall cell biology. And so the first thing that Suhita did, the grad student who found this data, was she knocked down CAPZB using an shRNA and showed that it promotes signaling similar to SAMD14. So just sort of showing you here, we can divide up cells between, divide up KIT positive cells by CD71 positivity, so kind of the low medium, which would be more the BFUEs, and then the high cells, which would be more CFUE associated. And so she can see that in kind of the low medium cells that when she stimulates with SCF, CAPZB knockdown has lower activation of ERK. And in the CD71 high cells that express EPO, she knocks down CAPZB and she sees lower activation of ERK. And so that's just quantitated over here. So CAPZB is promoting KIT and EPO signaling similar to SAMD14. And then we did a number of studies. I won't go into all these experiments, but CAPZB knockdown experiments also alters the cellularity of erythroid cells in culture, and shows that if you knock it down, you get more differentiated cells. These cells are also smaller, and indicating that this capping protein is coordinating erythroid maturation. And then finally, just kind of to show you what this assay looks like. Again, so these are all KIT positive cells. They're we have a gradation of CD71 positivity from low to medium to high, and prior studies have sort of shown that these CD71 low KIT positive erythroid cells are really more BFUE forming colonies. And our data also supports this. And then the CD71 high cells are sort of more CD71 high are CFUEs. And then there's sort of this transition zone in between of this CD71 medium. And so by flow, we can look at here on the y-axis is just phospho-ERK or phospho-AKT. And we can look at kind of the median fluorescence intensity at each of these at CD71 low, medium, and high. So SCF activity is highest in CD71 low, EPO activity is highest in CD71 high. If we knock down SAMD14, or if we express SAMD14, we see increases in SCF-mediated ERK phosphorylation. And then in the CD71 high, we see higher activation of EPO instigated ERK activation. And in the context of SCF, this is capping protein dependent. So if we knock out the interaction motif that is required for SAMD14 to interact with capping protein, it reduces the ability of SAMD14 to activate AKT and ERK, but it's independent in the wing of or in the ability of SAMD14 to promote EPO signaling. And so mechanistically, the future directions of the lab are focused on identifying how these different interaction domains are kind of have distinct abilities to promote specific signaling pathways. Both SCF and EPO are operating through similar intermediate signaling molecules, AKT and ERK, and yet SAMD14 is able to regulate their activities through different interaction domains. And so how this occurs is really an active area of investigation and one we're really interested in, because the switch from SCF-dependent signaling to EPO-dependent signaling is crucial for the development of the erythroid lineage. Yeah, so I've gone a little bit over time, but these distinct requirements for SAMD14 in promoting these distinct domains, the SAM domain and the capping protein domain, that are promoting KIT signaling and or EPO signaling is a really, really interesting future direction that we're interested in. And kind of putting it all together into kind of a model and bringing it back to kind of our original goal for these studies. We've identified transcriptional mechanisms and cell signaling amplifying mechanisms that are accelerating red blood cell production in anemia. Some of these transcriptional mechanisms are operating through enhancers that are very similar to the SAMD14 enhancer. And I think I've shown you that we can use it by using this approach. We can identify additional enhancers that may have erythroid promoting roles in the context of stress erythropoiesis. We've also linked many of these transcriptional, some of these transcriptional activators to cells, environmental sensing and cell signaling mechanisms, including SAMD14, but probably dozens of others that are on our list of transcriptional activated genes. And we think that these genes are really important for amplifying signals that are coming from SCF in the context of anemia, because they really need to accelerate erythropoiesis beyond what is already a very, very rapidly proliferating system, beyond what is normally there. And so these cell signaling intermediates can help with that mechanism because the exact same SCF response is going to be different depending on whether SAMD14 is there or not. So with that, I would like to acknowledge the lab and the people that did most of the work. So a lot of the single cell and transcriptional data was done by Yichao. He's here in this photo. And a lot of the SAMD14 work has been pioneered by Suhita, who is graduating soon and moving on to a career. She hasn't quite decided whether that's in academia or not, but support her either way. But they did most of this work. Also, we have great collaborators that have helped us with the bioinformatics analysis, both the transcription and the proteomics. Great core facilities here at UNMC, flow cytometry and genomics, and also think funding as well. And I'd be happy to take any additional questions that you have.
Awesome. Thank you, Kyle. That's great. Do we have other questions for Kyle this morning? Dean, I see your question there. Do you want to ask it? Or do you want me to
Sure.
ask?
Can you hear me okay?
Yeah.
Yeah. So I really like the kinetic data that you showed in the last bit. There's a lot of different patterns there that suggest that things may be more complex than the factors you've identified so far.
Mhm.
And one thing I noticed is that the E-box you showed in the SAMDH1 enhancer, it has a second E-box immediately upstream, right at the place where you did the deletion. So there's two tandem E-boxes. And that immediately makes me think of MICMAX, E2A, and unfortunately, it may be clock BMAL in the circadian patterns. So I'm wondering whether or not, especially maybe later in the time course, you think that MIC, for example, might be binding there and helping to expand those populations, but at the same time induce that specific gene pattern so that you can get sort of rapid restoration of the erythropoiesis.
Yeah, that's a great question. Whether there are ways to kind of parse out the sequence determinants that may be present at some of the enhancers that have differential kinetics to others. We haven't looked at MIC. That would be interesting to look at as well. But it looks like, at least just kind of from a broad perspective, all the EBOX GATA associated genes that we identified are not all of them, but the vast majority of them are coming up at that day 3 time point, but not day 1. So yes, that does suggest that kind of there are some other factors that are being induced kind of at probably the sort of pro-inflammatory stage of activation that are leading to the activation of these EBOX GATA.
Do you know how many have 2 E-boxes?
I don't.
Split it up that way.
I'm gonna look, though. That's a really good point.
Yeah. Thank you.
Yep.
Kyle, you had a bunch of other genes listed on that interacting. One of them was a gene I studied in graduate school, so I was super interested. It was SHMT2. You had like a whole bunch of them. Do you have any idea what the rest of those genes are?
No, we've tried to validate a few of the others with low success.
Okay.
There were a few. So which one were you?
SHMT2.
SHMT2. Yeah, we haven't looked at that one. There's an EIF4A1, and so we got that antibody and weren't able to validate that it's actually a real interaction.
Okay.
And so that's kind of been our strategy there, is if we can validate it, then we'll move forward with identifying what that interaction
it's possible these are like big complexes too, like I don't know.
Yeah.
Anyways.
Yeah, I mean, our proteomics collaborators have tried to kind of sift through it to filter out the crapome, is what they call it. So the junk that is coming down with lots and lots of IPs. And these might, some of them might be very weak interactions. It's also important to note that we're overexpressing SAMD14 with this HA construct in G1E cells. And so, some of these might be effects that are caused by kind of elevated expression of SAMD14 in order to kind of capture these interactions.
So to that point, Kyle, is it CMV? Are you expressing under CMV?
Yeah, yes.
Okay, because there's a guy here at the biochemistry department, Wes Sundquist, made mutations in the CMV promoter to make them more endogenous level expression. I have those if you want them, and I know Wes would let me share them. so that you could test your question of overexpression or not by using the same promoter, but just mutated to drop the expression. Anyway, just an offer.
No, that's good. I mean, we've thought a lot about that and had, multiple lab meetings kind of trying to get at, trying to assess that, using inducibles. The approach that we sort of settled on, it's in the paper, but I didn't present it, is that we sorted out cells that are really low GFP expressors.
Yeah.
And then we also sorted out the very high GFP expressors, and we did Western blots with both populations. And it does seem that the level of SAMD14 correlates with that, right? So the lower GFP has lower SAMD14.
So you already know the answer.
Yeah, and well, we know the answer for CAPZB, the capping proteins, and they are binding to SAMD14 in both contexts, but we don't know that for any of the others on the interactome list, whether they're interacting with SAMD14. But the delta SAM data, where we also did proteomics on that, and there's really nothing that the SAM domain of SAMD14 interacts with at the protein level. And so we have a very ambitious graduate student in the lab who's trying to identify what if not protein, what else SAMD14's SAM domain might be interacting with.
Cool.
Yeah.
Mark, you have a question in the chat. Do you want to ask?
Sure. But before I get to that, Kyle, have you looked at transient interactions with your SAM protein? Like, is it possible it's interacting with other proteins? It's just really brief.
Yeah, that's so to address that, we've done cross-linking experiments where we've cross-linked it and looked for really for those experiments we've been looking for self-interaction. There are several studies on SAM containing proteins that have shown that they can be very labile, have very weak dissociation constants. Some of them are the opposite. Some of them have extremely high dissociation constants, but we did some cross-linking experiments to try to address whether it was transient. We've also done some acute stimulation experiments with SCF to try to see whether any interactions were signaling dependent. And so far, we haven't really hit on anything interesting, but that we haven't really kind of abandoned those lines of investigation yet. Just nothing interesting has emerged yet.
Have you tried Halo or Turbo or NanoBret?
No, it's been kind of crude methods so far.
I gotcha. Cool.
What would you, I mean, if you were to pick one of those methods, what would you choose?
Turbo. NanoBret's also really cool, if you're familiar, but it's in vivo interactions that you can see in real time, which is nuts. But then you also have to know what proteins you're scanning for. So it's not like big data kind of a thing. But Turbo is instantaneous, and it labels all the interacting proteins that are within like 30 angstroms or something.
Yeah.
So it's really good. But it's huge, so it's hard to clone. And it might also disrupt, interactions.
Yeah. I mean, we'd love to do that kind of in a cell signaling response kind of assay to see whether whether a SAM interaction might be either coming on or coming off in response to signaling.
For sure. The question I asked in the chat was, have you looked at the cytoskeletal structure with your capping protein or with SAM D14?
No, we did with the CAPCB knockdown, we did. But we have not looked at the cytoskeletal changes that are kind of induced by SAMD14. That's another kind of probably a very important future direction to try to understand the mechanism for how SAMD14 may be promoting signaling and survival and all the and stress erythropoiesis in general.
Yeah, because I think there's like different subpopulations of AKT1, and one of them I think actually goes into the inner leaflet of the plasma membrane and connects to the, I think it's actin cytoskeletal structure. So when you said those two things, it kind of popped in my head.
Yeah, that possibly, yeah, that its interaction. Well, we know that its interaction with the capping protein complex is important for its signal promoting effect. Our models are really kind of whether that interaction might be altering cytoskeletal structure to promote signaling. It's almost like an indirect model, right? Its expression is not direct, it's not directly involved in signaling, but it's changing cytoskeletal structure in a way that promotes it.
Right.
It could also be just kind of a subcellular localization thing where it needs to be SAMD14 needs to be at a certain place in the cell, and that the capping complex is kind of keeping it in the right location.
Yeah, it'd be super cool to look at.
Thank you.
But great talk. It's really cool stuff.
Yeah.
I have a quick question. So there are many cases of the human hereditary anemia. Have you looked at have you ever cataloged or doing anything with the SNP, presence of any SNP that are associated with the genes that you have looked at it, or?
Yeah. If we take that list of 65, the anemia upregulated EBOX-GATA associated genes, a lot of them are associated in GWAS studies with hereditary anemias and with leukemias as well. And so there's several genes on that list that are those are really kind of the top hits in those gene ontology analyses.
Okay, thanks.
We do think we've also played around with kind of comparing our data to several other single cell sequencing data sets that have used chronic anemia models and other types of anemia models. Our gene list overlaps very nicely with other acute anemia models. So there's a model where they put mice in hypoxia chambers and then looked at transcriptional changes induced by hypoxia. And a lot of our genes are on their lists in the same gene clusters. Chronic anemias, we do see some overlap in some genes, but SAMD14 is not on those lists in general. And there's a lot less overlap. So once you get into the chronic context, there's a lot of things that are likely going on transcriptionally that are kind of not relevant to our kind of acute anemia models. But we think kind of the proximal effects. So like doing these acute anemia models, we really get at the immediate responses at the transcriptional level that we can study mechanisms of. But yeah, that doesn't always translate well into chronic models where there's a lot going on kind of at the multi-organ physiological level that we can't model with the acute system.
So initiation of the chronic anemia is different transcriptionally.
Yeah. I mean, that doesn't mean that the mechanisms that we see in acute anemia wouldn't be relevant to chronic anemias, right? It just means, that system has gone so haywire in the chronic context that those cells cannot mount an effective regenerative response.
So it boils down to the signaling between the SCF, the KIT, and EPO, even in the chronic case.
All right, awesome. Well, Kyle, you stimulated a lot of discussion. That was awesome. That was a great talk. Yeah. I'll just tell you one last thing, Kyle. I loved your single cell analysis. It was super thoughtful, actually. I thought it was great. And I think actually my experience is doing trajectory analysis with like progenitors that are all the same or cells that are not like very far apart is garbage. But that's my two cents. Anyways.
Yeah, no, that's that. I thank you. We put a lot of thought into it, for sure. We want to be able to get, I think more data out of it than what you see in a lot of papers, which is just differential.
No, it shows. I mean, actually, like considering like expression of your gene of interest, in different, in specific cells across different, I mean, I thought it was super thoughtful. And I do think like monocle and trajectory analysis in cells that are not like vastly different, like at opposite ends of the differentiation trajectory is kind of nonsense. I mean, even within the stem and progenitor compartment, I find it kind of nonsense. So.
I know.
Anywho, all right. Thank you so much, Kyle. That was great. Really appreciate your time and energy and commitment to this. And yeah, thank you all for attending as well.
Yeah, and we'll get you this video posted.
Okay, well, thank you for inviting me. Appreciate it, and all the great questions, certainly stimulating.